great. My name is Jacques Cohen, and I'm going to hurry on and introduce our first speaker from Edinburgh, Scotland. And I learned that Professor Telfer was the first PhD student of Roger Costin, which is an accomplishment. And she will talk about in vitro follicle growth and germ stem cells, an alternative approach to improving oocyte population. Thank you.
Okay, thank you very much. And we started the day looking at the in vitro growth of oocytes all the way through from primordial germ cells. And that was the mouse model. And we got the nice work from Yuji Hirao there. And the Japanese group have really been, at the forefront of developing these methods in the mouse. But of course, we would like to be able to apply this to humans. So what can we do in the human situation? So of course, the dominant sort of theory about primordial follicles and germ cells is that we have this store of primordial follicles, and that's the resting. Kutlik-Oktay spoke about this and how they might accumulate DNA damage, but then they'll start to grow and develop into preantral, late preantral, and antral follicles, and in the human, which is a monoovular species, only one of these would reach ovulation. So, of course, Kutlik also said that 99.9% of these germ cells are destined to die. So the question is, can we save these germ cells and can we utilize them by growing them in vitro all the way through to the point where we could mature them and fertilize them and get healthy embryos? So, of course, for those of you who work in the area of fertility preservation, and Kutlik has developed a lot of the techniques involved in fertility preservation, the young women that come to the clinic are offered the opportunity to have pieces of their ovarian cortex cryopreserved. And when you look at these tissues, then, they're predominantly primordial follicles. Majority of the follicles in that tissue are at the primordial stage. Now, of course, the reason for the cryopreservation is often for the tissue to be reimplanted. But that cannot happen in some cases because of the presence of malignant cells, so you can't transplant them back. So what do you do with this tissue? Then, of course, the aim would be, could you take these cells, these primordial follicles, and grow them all the way through to the point of maturation and fertilization? And that's a challenge, and we know from the mouse that it can be done, but we have the challenge now in the human. And this is an introduction to the first mouse that was born through growing a primordial follicle all the way through to the point where the oocyte could be matured, fertilized, and the embryo put back into a pseudopregnant female. And this is Egbert, who was born in the Jackson lab. And Egbert really set back the whole process of follicle culture in humans because there was worries that this mouse was actually epigenetically abnormal. There was no evidence for this other than the mouse was fat and lazy, but it was a one mouse. And you do get some mice that might be fat and lazy, but this was particularly fat and lazy. And when Egbert died, Egbert had lesions in almost every single tissue that was looked at. So it was a concern. But the EPIG group transformed. They changed their media formulations, they changed their methodology, and then they've been able to get lots of mice that could be developed from primordial follicles in vitro. And of course, we now know through the work, the Japanese group that was discussed this morning, that you can actually take them all the way through from primordial germ cells and get the development of embryos and indeed live young, which appear to be normal and healthy offspring. So then applying this to a human situation is a complicated process. And making an egg is a complicated process. We know that that oocyte then has to be held in meiotic arrest. It has to be activated to grow. It has to acquire meiotic competence as it goes through its development within the follicle. And it has to undergo transcription. There'll be transcriptional repression towards the end of development. And of course, genomic imprinting at each of the appropriate stages. So this is a complicated process, and to try and recapitulate the whole process in vitro is indeed a huge challenge. And a huge challenge is to get a normal, developmentally competent oocyte. So we've been working in culture systems for many, many years, starting out working on the mouse and then moving on to the cow and to the sheep and to the pig, and now working on human material. And what we've been looking at is developing a multi-step culture system, similar to the systems that have been described for the mouse, but that would be appropriate for human oocyte development. So we've been optimizing the growth from the primordial stages and then supporting the development of the growing follicles, then looking at the final stage of oocyte development, and of course then testing whether these oocytes are normal and are capable of being fertilized, which we're not allowed to do at the moment, but we're hoping that we will be allowed to do in the near future. So how do we do this? We are in a position where, with clinical collaborators, Hamish Wallace and Richard Anderson in Edinburgh, we can get a whole range of human ovarian tissue. So we get them mainly through at the time of caesarean section. So women are recruited and they're, they give their informed consent for a small piece of their ovary to be taken at the time of caesarean section. And we also get them through gynecological procedures and fertility preservation program. So that means we can access a range of tissue from very young to older material, and we get it fresh, or we get material that's already been cryopreserved through the fertility preservation program, and then it's donated to us at a later stage. So this is what our tissue looks like, and this is what we're working with. So we get our ovarian cortex and we prepare it in such a way where we remove all the underlying tissue and we only have the cortex that has predominantly primordial follicles. And we would say we prepare this micro-cortex for culture. And then when we put it into culture, you can see the follicles have initiated to grow, and this is very quick. It's within 6 days you get follicle initiation. Now, I'm sure most of you are sitting here, many of you might say, that's too fast. That wouldn't happen. How can that happen? And I personally thought that too, until I actually saw it. But now we understand the molecular background to that. And the work of Erin Shui showed us that when we prepare this tissue, we're interrupting the hippo signaling pathway. And that's leading to a greater activation of these dormant primordial follicles. So there is a mechanism surrounding what we were observing many years ago when we were culturing human tissue. And when we culture them in this short period of time, we can see multilaminar follicles. And I know the cynics, and I'm probably the most cynical person you'll meet, and the skeptics, will say, well, they've started to grow, and you're getting them, that's why you get them in a short period of time. But that's not necessarily the case, because if we go back to reculture tissue where there's only resting follicles, we know that this still grows in that period of time. So this seems to be a particularly fast rate, but I would say it's a rate of development without the normal negative controls that you get in vivo. So it's not necessarily a bad thing, but the follicles can grow to pre-antral stages in a reasonable time, and more importantly, in a workable time to grow them in vitro. So when we get these follicles and they form the sort of multilaminar stages, the pre-antral follicles, we then dissect them out. And we heard earlier this morning about the use of collagenase. We do not use collagenase in the human follicle preparations, because in our hands, collagenase has a detrimental effect to the development. So we use microdissection to remove the follicle with the theca layers. So they have theca layers. And that's important for the human follicle to maintain its three-dimensional configuration. And when we grow them in the presence of activin, and activin is important to maintain oocyte somatic cell interactions, and a very, very small amount of FSH is also important, we can get the development of antral follicles in this culture system. So that's the human follicle culture system in our first step, activation of primordial follicles. Second step, isolating the follicles, because if we leave them all together, they start to talk to one another. There's communication which causes some of them to degenerate. So to optimize the chance of them surviving, we grow them individually. And we grow them individually, and then they'll form antral cavities. So then our next stage was to develop the oocyte itself, to get it to a point where we could see if these in vitro grown human oocytes were capable of in vitro maturation and resumption of meiosis. So when we were starting this process, we decided not to go with completely culturing the complete follicle, antral follicle, for a long time. And some other groups have done this. And some of the domestic species work that's been developed by Helen Picton, for example, they culture the very large antral follicles. But we thought, well, we're more interested in this oocyte and its surrounding cells, because a follicle has the function of oocyte development and also endocrine function. And for the purposes of oocyte growth, we don't really need this endocrine function of the follicle. So we isolate this part of the follicle. We take the oocyte with its surrounding cells, and then we grow that for a further period of time on these membranes, similar to the material, the co-star membranes, transwell membranes that was described in the first talk today. So we culture those for a further period of time. And what we find is that once we have that antral formation and we grow these complexes, we can, of the oocytes or the follicles that survive in our culture period, and we don't get as high survival rates as Yuji was saying in the talk this morning, but we get about 30% of the oocytes that do survive can reach metaphase 2. Now, of course, we need to look and find out if they're completely normal, and that's work that's ongoing. And this is just some images of the in vitro grown oocytes, all the way from the primordial stage up to the point where they can reach metaphase 2. And you can see here the spindle, you can see the polar body. Polar body is larger than you would normally see, and we don't know why that is. We think it's some kind of physical condition in terms of, how they've been growing and then how they separate. But that's something that we would need to look at in more detail. But they do reach metaphase 2, and there's a proof of concept now that we can get them to that point. So this multi-step culture system supports human oocyte growth and development. Of course, optimization at each step is required, and some of the mouse work will inform us what we might be able to improve upon in getting these human oocytes to develop. We need to test them further, of course, for how normal they are. And the ultimate thing, of course, is their fertilization potential and development of embryos. Clearly, if they produce live young, but that's a bit early to go down that path. And then whether the technique can be used in young girls, because most of the ovarian cortex that's stored in fertility preservation clinics is from prepubertal girls. It's from the young girls, because older women have other options for fertility preservation. So the question is, could you use this technique to grow and develop tissue from the prepubertal girls? So we did a study where we looked at the patients that we had in fertility preservation, and we had a range of patients with a range of conditions and ages, and we divided them up into the prepubertal and then just around puberty, and compared them to tissue that we'd obtained from older women, from women that were post-pubertal. When we grow the tissue from the prepubertal girls or around puberty, they activate, the follicles start to grow. It's absolutely fascinating tissue, because you'll find nerves growing, you'll find blood vessels starting to develop. It's really highly active tissue, and that's just showing you some of the imaging of these follicles growing, and obviously other processes going on in this tissue. So they do grow, they start to activate. But when we isolate the growing follicles and then try to grow them in the second stage of development, what we find is that there's a difference according to the age that the follicles have come from, the age of the girl or the woman that the follicles have come from. So if we look at the youngest ones, between 3 and 10, when we start to grow them, the isolated follicles, they do not grow as well as we would see in the older age group, so the 25 to 38. And then 12 to 15, they grow better than the youngest group, but certainly not as well as the older, or the 25 to 38 years. So there's clearly a difference in terms of their ability to grow and develop according to their age. And that's something that we're very interested in. So conclusions to this part then is that we can get follicles to initiate growth at all stages to the secondary stage. The follicles from the younger girls, they grow slowly and they show little oocyte growth. So there's a difference in terms of whether they come from prepubertal or postpubertal. And we need to look at that in more detail and try and adapt the system to see if it is possible to get them to develop further. And of course, within that age range, there's a difference. So age is important in terms of the follicle's ability to grow in vitro. And that does have implications for fertility preservation, because the question is, well, what's different about these oocytes or follicles that come from the young girls? What can we see any differences? And we've published this work. This was published in Human Reproduction. And when we did a detailed morphological analysis of the tissue that had come from the younger girls, what we found was that, in fact, there was a whole population of oocytes that hadn't really been previously described. And these oocytes were larger. We call them the big fat oocytes. And they also, some of the times, when we looked for a germinal vesicle, we couldn't see a germinal vesicle. So there was a population in the younger girls that, was quite a high proportion of the oocytes that were there, where, of this morphology. But that decreased as with age. And in fact, in the adults, you never, ever see them. You don't see them. So you see them in the prepubertal girls, you see them around puberty, but by the time adulthood comes, you don't see this population of oocytes at all in the human ovary. So it's important for us to know about these and to start to identify ways of perhaps removing them from the culture system and maybe getting the other follicles to start to grow. Because it may be that there's clearly a mechanism in vivo where these are taken out before adulthood. So this is a model system where we can start to understand mechanisms of oocyte development. Other groups have achieved meiotic maturation from growing preantral follicles in vitro. So the group of Teresa Woodruff has shown this with isolated preantral follicles. And we're getting closer to testing the fertilization potential of these human in vitro grown oocytes from primordial follicles. But this system can also be used to address another question, not just whether they'll grow from the primordial stage, but can you actually make new oocytes from another cell type? And again, the first presentation this morning discussed making oocytes from primordial germ cells. So what we're talking about here is making oocytes from female germline stem cells, which is indeed a highly controversial area, and I'll spend the next part of my talk giving some details of what we've been doing in this area. So the question is, can you build gametes? Can you make them from a cell type that's present in the ovary and get them to develop down the pathway of oocyte development? Just like the Japanese group, Hayashi and Sato, have been able to do with the induced pluripotent stem cells and the embryonic stem cells. So we've been looking at isolating and characterizing a putative oogonial stem cell population from the human ovaries. And this builds on work that was going on for a number of years in the laboratory of John Tilley in Harvard at the time, or in Mass General Hospital. And John Tilley had been working on a mouse model, and it generated a tremendous amount of criticism, and I was probably one of the critics, or I was one of the critics of that work. And then in 2012, a paper was published by the Tilley group where they isolated a population of cells from human ovaries, adult human ovaries. Again, it's a tremendous amount of criticism of this work, but essentially what they found was that a population based on isolation using DDX4, an RNA helicase, also known as VASA, mammalian VASA, and they said that, okay, we can isolate them on the basis of this DDX4, which in itself was controversial, and these cells are capable of division, which they showed using BRDU, so dividing cells that had a germline marker. So we've gone on to look at these cells and to ask the question, well, you know, if these cells are DDX4 positive, we should be able to see these cells in sections of the human ovary, because DDX4, we believe, is exclusively in the germline, and everyone accepts that DDX4 is expressed in the oocytes. So when you do an immunocytochemistry, you find that you have lots of staining of DDX4 in the oocyte. But if you look closely and you go through your sections, and this is a laborious process, because what it means is you have to go through sections and sections and sections and look and see. And what you find is, you find that there's small, I need to go back there. You find that there are small cells. Yeah, okay. It's lost it. There's small cells here, and I should be a bigger view, but it's some reason it's not showing up. There's small cells that are indeed DDX4 positive. So if you go through the human ovarian cortex, you will see these cells. So the question is, well, what are these cells? And we believe that these are the cells that had been isolated by the Tilly group, and we've gone on to try and isolate these cells also. Now, again, many groups have published papers saying they cannot isolate them. And I would say it's not the easiest process, but we can isolate a population of DDX4 positive cells using the FACS sorting methodology, but we also use a size discriminator. So we would collect the small cells because we believe that these would be the ones that would be the potential or putative ogonial stem cells. And the larger ones, of course, could be small oocytes. We don't know, but we're looking at those cells. And these cells, when we do PCR and we also do Western blotting, the positive cells have DDX4 and the negative cells don't have DDX4. So there does seem to be a discrimination based on that. Now, of course, there's an argument about whether DDX4 is surface or not, but leaving that aside, these antibodies do seem to isolate separate populations. And when we take these populations of cells, the cells that have been isolated, and we mark them with a green fluorescent protein, and then inject them into our culture system, into these ovarian cortical pieces, we can actually see a progression of these cells. So you can see when they've been injected, these cells are small. So this is a chromogenic marker for the GFP, so you see the brown cells there. And then you see larger cells. The brown cells have become larger. And they're starting to, you know, it looks like they're associating with some of the somatic cells that are around. And then you can see here, they have associated with somatic cells. And then in this image here, this is I'm sure most people would say this is an oocyte, and these are granulosa cells, and it's a primordial follicle. It looks like one. But the cells that make up the oocyte have come from the cells that have been injected. So it looks as if these cells do have the potential to form what appears to be an oocyte. And of course, their further development and characteristics is something that we're in the process of studying. So building gametes is complex, and it has a lot to do not just with the cells themselves, but also with the somatic cell support that they get. And that's something that we really need to study in more detail. What feeds off these cells? How do they communicate? And we're isolating a population of cells within the adult human ovary that seem to be capable of making an oocyte or follicles in vitro. And we don't know if they're a true germline stem cell. There's a lot of work to do. But the in vitro model will allow us to assess that. And I'll just quickly, I've got a few minutes, I'll just quickly finish off by some evidence then that these cells may be acting in vivo. And we don't know if it's the cells, but we've found a situation where it looks as if we have more follicles than we should have. So something's happening. So can new eggs be made in vivo? Now, what we've done is, and this was an entirely unexpected result, we had tissue from patients that had been treated with ABVD, and we had tissue from patients that had been treated with OPA-COPDAC. So both treatments for lymphoma. And we then did a detailed morphological analysis of their primordial follicle, a non-growing follicle population. And you can see here in the red, this is the black dots are the normal lines. This is the number of follicles that we'd expect to see at each age. And this was also described by Kutlik. And we've done a lot of modelling with Tom Kelsey and Hamish Wallace. And then you can see here the ABVD, which is a non-sterilising treatment. We're seeing an increased number of primordial follicles. We don't know how to explain that yet, but it looks like this treatment is causing something to happen. Whether it's related to stem cells or not, then we have to really look at that further. But the other thing that we're seeing with this tissue is not only are there more follicles, they also look like the follicles that I described to you that we saw from the immature ovary, from the pre-pubertal ovary. So it's almost like whatever's happening with this treatment, the ovary's gone back to an earlier stage. And we don't understand the mechanism, but we think that this is a really important observation and one that should be followed up and studied in more details. So, of course, there could be other mechanisms to explain this than just the germline stem cells, but it's one that we are hoping to pursue in more detail. So just to finish off then, the field of adult germline stem cells is new, and the concentration of effort in the ovary, in the adult mammalian ovary, has been in doubting their existence. So we've got a big body of work that doubts their existence and has proof that they don't exist, which is actually quite difficult to get. But there is positive proof for their existence in a range of species, in bats and cows and pigs and rats and rhesus monkey and lemur. Now, their physiological significance is still unknown. But I think it's important for us, if we want to move forward in terms of developing new strategies for infertility and treatment of fertility and fertility preservation strategies, we need to understand the population of cells that do reside within the ovary. And it's only then that we could move forward. So I'd finally like to acknowledge the people that do the work in the lab and my collaborators in Edinburgh, and of course the funding body, the Medical Research Council, and all of the patients who donate their tissue to us. And thank you for your attention.